Thank you for joining me for the 14th episode of the Doc Talk with Liz podcast. Our guest today is Dr. Dan Mandell. Dr. Mandell is a leading expert in synthetic biology, protein engineering, artificial intelligence, and biological and medical informatics. Together with Dr. George Church, he leveraged GROs to produce the first proteins with stability and function dependent on non-standard amino acids. Dr. Mandell completed his PhD at University of California, San Francisco, his master's at the University of Edinburgh, and his undergraduate degree in symbolic systems from Stanford University. So, hi, Dr. Mandell. Thank you for joining us today.
Thanks, Lizzie. It's a pleasure to be here and to see you again.
Yes, absolutely. So, I know that you're doing so many amazing things now. You're the CEO of a biotech company, and you're like doing great research with GROs. But before we kind of get into the work that you're doing now, would you mind telling us a little bit about your journey into science, like with your childhood, if either of your parents were scientists, or kind of how you developed this passion for science?
Yeah, sure. Yeah, my parents weren't scientists per se. My dad worked in the medical field. He was a child and adolescent psychiatrist, and my mom was in education. My dad actually was actually pretty jaded in medicine, and actually often discouraged me from going directly into clinical practice if I ever even was going to consider that, and kind of suggested if you're going to get into health or medicine, do it through technology or science. And I think I did have a penchant for trying to figure out how things work, and making and breaking and building things, usually breaking things. And I think I got into this sort of through the computational side. So I grew up in the 80s, and so it was like, we were just sort of the kind of thing where a kid could pick up basic programming and mess around at home, and actually got really into computer animation kind of early on, and now we're into the 90s, and but I'm not artistic enough to make. I wanted to work for like Industrial Light and Magic, and make special effects for George Lucas, and it just I was able to play around with it, but didn't really have the artistic talent, but sort of got interested in what was going under the hood. And so as I got into college, I got pretty heavy into computation, and never actually was really good early on with my hands actually in science, in like biology class, for example. And so I definitely came at it through the computational side, and I was at Stanford for my undergrad and did this degree called symbolic systems, which is a major that's kind of like cognitive science in other schools, but it's kind of a strange mishmash of it's really computation, philosophy, psychology, linguistics, and logic. And you kind of end up focusing it on something. And what I ended up focusing on was starting to try to model biology with these computational tools. I was really inspired by a class I took with Russ Altman at Stanford, which I believe was my junior year. And as I got into the end of college, I started looking for ways to model biology in ways that we can't or couldn't at the time with these tools that were emerging in AI and machine learning. And this is now kind of early 2000s, and so we as a field were still not really well accepted. We were just being able to sort of do things like voice recognition was going from embarrassing to not so embarrassing, but people didn't really think much of it. And so I ended up doing a master's degree at the University of Edinburgh in Scotland. There actually was supposed to be this program between Stanford and Edinburgh that didn't end up happening, that would have sent me there and paid for it. And so that went away. But in process of preparing for that, I had set up this collaboration between a really talented AI professor at Stanford and a really interesting professor of psychiatric genetics at Edinburgh, where we were going to model sort of transcriptional networks involved in schizophrenia in a mouse model. And again, the data never really emerged on the experimental side. And this is something I guess I started learning early in my career, is the challenge of relying on somebody else to provide you with data. But I ended up getting access to a really interesting other data set around the postsynaptic proteome and protein-protein interactions that are involved in neuroplasticity. And so I did a project there trying to use machine learning tools to predict protein-protein interactions in this postsynaptic proteome from first principles, so really from primary sequence. And it was honestly an absurd project to try to do at the time. Like, it had really no chance of success. And it's really funny to me because now it's been 20 years later and you've seen this fantastic results recently from DeepMind and from David Baker's lab in Seattle, where this is actually now, you wouldn't quite call it a solved problem, but it's incredible progress. And we can now predict interactions from primary sequence from so many proteins. A lot of this is also due to advances in the templates that are available to us from crystallography, but also there's been these tremendous computational advantages. So long story short, this project was ill-guided, and it made for a nice master's degree in the sense I could do the work and perform an analysis, but it did get me really excited about actually how proteins work.
Awesome.
And so that was so that kind of is what made me end up going to UCSF, where I did my PhD. And I wanted to really understand, what are the sort of biophysical principles that govern protein folding and protein interactions.
I was going to say, because like what you're talking about with your undergrad degrees, like really interdisciplinary work, and biophysics is something that really interests me. And I feel like kind of drawn to it because of the interdisciplinary nature of it. I think like it's not just like you're looking at one thing. I'm personally really interested in like the 3D genome and that kind of research with biophysics, but I think that it's a really interesting like combination of potential math modeling with just like regulating transcriptional networks and that kind of thing. So I think you make a good point about that. And the other thing I was going to touch on was when you were mentioning like not being very good with your hands. I was working in lab this summer for iGEM, as you may know, and I contaminated so many samples. I was getting like so frustrated, but I kept going on with it. And I'm like, in the future, I think I want to keep doing this, but maybe like not all the time. So I'm thinking like maybe computation would be an interesting pathway to go down.
Yeah, it's an interesting point. It's kind of unburying the lead here. It's funny because my postdoc advisor, George Church, really he doesn't believe anybody has bad hands. Everybody can have good hands if you work at it, and you would too, right? It really is. I mean, some people do take to it faster, but it's like anything. If you care about what comes out the other side, you'll get there.
That's true.
But we are entering. I guess we have entered into an era where you can very much do both. You can do computation, you can make things, you can test them. And in fact, there's a huge advantage to that. Coming back to this notion of being able to create your own data and not have to rely on someone else to make it for you. And there's a fine line to be walked there, because there's so much important work that comes out of collaboration. But early on in your career, the more you can rely on yourself, the better in many senses, because you don't have a lot of leverage in case something goes off the rails with somebody else. And that was something I did have the luxury of doing when I did my PhD. I was in a laboratory. I worked with Tanya Krotemi at UCSF, and she did her postdoc with the aforementioned David Baker at UW, right? So from this computational design lineage, right? And so Tanya's lab was really great because we did have this ability to learn how to do protein design, learn structural biophysics, use tools like Rosetta, which is the software package that came from David's lab initially that many people around the world now co-develop, and then make the proteins and test them, right? And I did get to come full circle. It took some time, it took years, but you got to the point where you could do that. And that was really, really exciting, right? To be able to, design something in silico, and then be able to get into a laboratory and make it and test it. And so this really did kind of, I think, build a lot of passion in me around this field. But I was also kind of struck by this limitation that you see in proteins, right? That oftentimes, you wish you had a slightly different chemistry that you could place in a certain position, or a slightly different geometry or sterics. And we have these 20 amino acids that we can work with. And of course, everything in life that you see around us is built from them. There's certainly no shortage of things we can make. But when you switch from sort of evolution of living systems to evolution or engineering of something with a specific purpose on a small time scale, opening up that sort of toolkit, as it were, adding more amino acids to our alphabet, would go a very long way from a design perspective, right? And that was something that always kind of struck me. And so after that PhD, I came out to Boston and did that postdoctoral work with George at Harvard Med, and him and his lab were just finishing these new organisms, these so-called genomically recoded organisms, that let us go beyond that 20 standard amino acid alphabet. And as like a protein design person, this was just an epiphany. I was like, my goodness, now we can really, anytime I wanted a trapezoid instead of a square, we can do that, right? So this was super exciting. I was lucky enough to, speaking of sort of developing, your hands and whatnot, to start a project with a number of really talented scientists in the lab, most notably Mark LaJoy, who was the individual who really led the effort to create that first recoded organism.
Right.
And so Mark really went a long way to teaching me the tools for doing what we call recombineering, the process of modifying the genomes of organisms, so that I could further the project along and do experimental work and even continue it after he actually, ironically, went on to work with David Baker in computational protein design. So we very much sort of crossed the paths there, and he certainly taught me a ton. I hope that I taught him something too, and he's now CEO of his own company that spun out of a company that's spun out of his work from the Baker Lab in a sense. So there's a lot of interesting sort of personal stories here, but it was the point where I would say, at least got decent with my hands and was able to not only do the computations behind some early I wouldn't really call it proof of concept, but I guess the kind of early work in non-standard amino acid engineering of enzymes that led to us gaining confidence that we could do this in general, both doing the sort of the computations and the experiments for that. So anyway, that's sort of the story of kind of how I ended up with George and Mark, and I can get into more into the company, but maybe I'll pause for a second and see if anything there requires further comment.
Well, I'm just kind of curious. I mean, obviously I've read the biocontainment paper because that's how I first got connected with you through asking you some questions about that. I'm kind of curious though, as if the reasoning for why biocontainment was the first application of using the biphenylalanine. Is there a reason like biocontainment specifically was the first thing looked at, or is it just kind of for, again, like more testing things? And
yeah, that's a great question. Yeah, there's actually a real answer to this. So, and actually it's probably worthwhile just for a moment, sort of demystifying what we mean when we say to recode a genome, what that actually means, right? So what we mean by that is really, I guess this still sounds like jargon, but we say like genetic code reassignment. So to bring that down one level more concretely, pretty much all of life shares the same genetic code. By genetic code, we mean when you have 3 consecutive nucleotides in a DNA sequence, like, ATG, whatever, TAG, CAA. There are 64 possible ways that any 3 consecutive letters of 4 possibilities can be combined. And so in pretty much all of life for the last, 3.5 billion years, that genetic code has been conserved. So inside of you, inside of me, inside of corn, inside of sloths, ATG means the same thing usually, and AAA means the same thing. And there are evolutionary, reasons for that. But there are 2 important features of that code, sort of beyond its endurance. One of which is that the code is fully specified. So there are 64 possibilities, and all 64 possibilities are spoken for. There are no sort of open slots in the genetic code. There are, however, the second property is that there are, however, redundancies. And so there's more than one way to say leucine, right? There's more than one way to say alanine, and in fact, there's more than one way to say stop, right? So there are 3 stop codons, TGA, TAA, and TAG.
Yep.
So one of the things that, I guess Mark and his colleagues noticed when they went about making this first recoded organism is that the TAG stop codon would be the simplest to remove from the genetic code because there were the fewest instances of it. There were 321 instances of it. And so the way they went about this was to sort of break those 321 instances into 32 different strains, each of which they changed 10 of them, right? And then so you have 32 strains, each with 10 changes, and then you can use horizontal gene transfer to turn those 32 into 16 and into 8 and so forth, until in the end you have a single organism with all 321 changes. And so that is how they made the first recoded organism. And what that means is now there are no instances of the TAG codon anywhere in the genome. But what it also means is there are what we call translational machinery in the cell that recognizes that codon for some kind of meaning, right? So if it's alanine, there is a synthetase and a tRNA that recognize the AAA codon to install alanine. In the case of the TAG codon, there's what's called a release factor that recognizes that TAG as a stop codon and that terminates protein translation at that position. Well, now there's no more TAGs anywhere in the organism, right? And so we can delete that release factor. So this is kind of a big deal because now you could, in theory, what we say, reassign the meaning of that TAG codon to direct the incorporation of a new amino acid instead of stop, right? And that's exactly what we did as a group, is to then install new translational machinery. And again, this is a synthetase tRNA pair that has to be orthogonal to everything else in the cell because you don't want to be putting standard amino acids into those positions. But now you can decode with this new translational machinery TAG as a non-standard amino acid insertion site. And so that amino acid insertion site is now present nowhere in the organism except where you put it in a protein coding sequence. Okay, so coming back to your question, I apologize for all the background. When you remove a codon from the genome, not only does it have the impact of allowing you to reassign it to direct the incorporation of amino acid, but it also makes the organism resistant to phages and viruses, right? And the reason why is that these phages or viruses require your cells to make their proteins, right? Which means they share the same genetic code with you, like everything else in life, and depend upon faithful recapitulation of that code in order for them to trick your cells into making their proteins and form new phages or viruses that will then come out and reinfect. So when you remove that TAG codon from the genome, you already get partial resistance to some of the most common phages. What's coming next is we're going to be recoding multiple codons in the genome of these organisms. And in fact, we have a new organism that's nearing completion, which will have 7 codons reassigned in its genetic code. Wow. And when that organism is done, it will be impossible for any naturally occurring phage to replicate in this organism. So you're going to have what we call a multivirus resistant organism. So this is actually really beneficial for biotechnology. It's not all that uncommon for a massive bioreactor or fermenter that's producing an important product, let's say it could be a therapeutic, to be contaminated with a phage or a virus, upon which you could be shut down for months and lose millions of dollars. But even more importantly, in some cases, patients have suffered because there's only one place that's making this drug and it's shut down, right? And that actually has happened in a fairly notorious case several years ago. With these organisms, that would effectively never happen. So that's a really important feature, or at least kind of a nice feature to have in any production organism. On the other hand, you could be forgiven for wondering what would happen if one of these organisms were to escape into the wild, right? Yeah. Is it possible that it could outcompete, naturally occurring microbes that occupy some ecological niche, right? Now, most scientists would say it's unlikely. There is no extant example of some, microbe being released into the wild and taking over, for the very reason that when we engineer these microbes, we're drawing so many resources away from what they want to do, which is reproduce, into producing something generally, right? So we've generally made them sicker, but it doesn't mean it's impossible, right? And so the answer to your question is, as we go to build these recoded organisms, we wanted to, George likes to say, build the seatbelts before the car, right?
Right.
We want to have a safety mechanism built into these organisms such that if they were ever to escape, they couldn't reproduce in the wild. And so whatever advantage they might have by these multi-virus resistant capabilities would be irrelevant because they couldn't survive in the wild. So that's why we did the project. And the project that you're referring to was, well, maybe, yeah, why don't you talk about it? And you, I don't want to, speak too much. So if you want to speak to it, I'm happy to answer any questions about it. But I think hopefully that answers the question of why we did the project.
No, yeah, I think that's really interesting because, like as you obviously know, there are like so many different kind of mechanisms, I guess you could say, for biocontainment. The ones that we kind of focused on a little bit more were more so toxin antitoxin systems, but Oxytrophy is, like you said, really interesting. I think the other thing that I was going to ask you, just to clarify for anybody else, was like getting the organisms to survive. You have to constantly supply them with something for Oxytrophy, right? And that is like the kind of bug that you guys were creating. It was an E. coli and it was an Oxytroph. So what did you supply to that to keep it kind of able to survive in lab?
Yeah, great question. So the approach that we used, we called synthetic oxytrophy. So oxytrophy in general just means the state of needing something else to survive. And so, humans are oxytrophic for certain amino acids. Some we can synthesize and some we have to eat, right? And so what we did with these organisms was recognizing, hey, if they were to escape into the wild and they could encounter this amino acid or metabolite or whatever you made them oxytrophic on, it's not a good biocontainment mechanism, right? Because they can escape. And so the sort of reference that comes up a lot when we talk about this is actually Jurassic Park. Because in this movie, among the safeguards that failed to keep the dinosaurs contained, they engineered the dinosaurs to be dependent on the amino acid lysine, right? The amino acid lysine is one of the standard 20 amino acids, and you could find it, pretty much anywhere in nature, right? And so big surprise that this doesn't actually work. And so when we did the project, we had to think fairly carefully about this, right? We needed to pick an amino acid that doesn't exist in nature. We also needed to make enzymes depend on that amino acid that operate on substrates that couldn't be found in nature, right? Because even if you made the enzyme dependent on an amino acid that can only be found in the lab, if the product that the enzyme makes could be scavenged from somewhere, right? You could bypass the oxytrophy, right? And the organism can still survive. So we had to think about a lot of things, like picking an amino acid that's really can't be complemented by anything, picking enzymes whose function can't be complemented, whose products can't be complemented, and so forth. So we really tried hard to think about that. Obviously, you can never be 100% sure you've considered everything, but we did as much as we could as far as proving out that this containment strategy is sound. And so that was sort of the inspiration behind it. Now, the way we did it was to combine these two worlds of computational protein design and these genomically recoded organisms, because, okay, now we can make proteins with these new amino acids. Well, which ones should we make and how should we design them, right? So the strategy that we picked was to basically take a number of enzymes that are essential for life inside these bacteria, and then try to use design to sort of imagine like the protein, imagine the enzyme's kind of we'll call it a ball, right? In the core of this enzyme, let's put the amino acid there. So it's really essential for folding. But on top of that, pick designs such that you have to really modify the shell of amino acids around that non-standard amino acid so that no other standard amino acid could complement it. So let's say you accidentally stuck a leucine there or an asparagine or something else. All of those would be so unfavorable, the protein would still unfold and you wouldn't be able to sustain life, right? And so in the end, we did a lot of designs, tested a lot of different enzymes. We were able to find combinations of designs of particular essential enzymes that when you withdrew that amino acid, the enzymes didn't fold and the organisms could not survive up to, we can test up to about 1 trillion organisms in the laboratory. That's about as high as we can go through the plate-based escape studies we were carrying out. And we did other things too, like look for, we would call it environmental supplementation. So let's say when bacteria lyse, they will release all of their metabolites into the environment. And is there anything in that, lysis soup that you could scavenge and be able to survive? And so we grew our organisms in that kind of milieu and showed that they couldn't survive. We also tried to carry out some very aggressive horizontal gene transfer and see, like, could you overwrite these safeguards in the genome of the organism by transferring genetic material from a related species of E. coli? And basically what we found was if you put enough of these containment mechanisms in, which ended up being three enzymes, three redesigned enzymes, either what would happen is you would only overwrite one or two of them, in which case you still had containment, or you had to transfer the entire genome, right, and overwrite the genome of the recoded organism, at which point you no longer have anything you need to contain because the genome is gone, right? So for everything we tried to break the system seemed to work. And so ultimately, we became convinced that, like, hey, we actually can do this. We can take these computational design tools with this expanded amino acid alphabet and engineer proteins whose folding and function critically depends upon this expanded amino acid alphabet. And so, jumping forward, 5 years after this project is really when, having kind of drawn that level of enthusiasm, that we thought to ourselves, well, what are some, really outstanding critical problems in protein therapeutics that we could potentially address with this expanded amino acid alphabet? And that was really the genesis of GrowBio, our company.
Gotcha. So you guys work more so on like protein therapeutics now rather than like biocontainment mechanisms. That's correct, right?
Yeah, and from a product standpoint, the company focuses on making protein therapeutics, really trying to identify which indications, which diseases have a really pressing unmet need for which we don't really see a way to solve it using proteins made from the standard amino acid alphabet. It's not trivial to add a new amino acid to the alphabet, and we have actually built some really exciting infrastructure in the company to accelerate that process. But we need to think carefully, again, about focus and what we call chemistries, which NSAA, non-standard amino acid chemistries, are worth the effort to put into the platform, because there is some pressing unmet clinical need that really can't be addressed without it. And we do a lot of our strategic thinking is trying to answer that question.
So if I can ask, is your favorite amino acid the synthetic amino acid?
I don't know if I have a favorite. Yeah, I know. Yeah, they're all beautiful in their own way.
Okay. Good answer. I had another question for you also, with like you being the CEO of GrowBio. I'm curious because I've heard of this before, and of course I'm early in my career still, so I haven't encountered it too much. But why did you kind of go into industry over academia? I'm just curious because I've heard of like differences between the two, or if there was like a specific reason, or if it's just kind of because you were so passionate about like working on this in your postdoc that it was a little like more natural, or?
No, it's a great question. And I think, for many of us, I'll speak for myself, but for those of us who've gone this route, I really think it comes down to sort of the natural fit for which of the day-to-day activities of the job you find, sort of most compelling.
Right.
This is a question I struggled with a lot, or I should say wrestled with a lot during my sort of the middle of my postdoc when, we had done the biocontainment project and we'd had some success there, and it wouldn't have been unreasonable for me to consider, trying to push for an academic career. And really what it came down to for me was the kinds of, again, day-to-day work that you find, most interesting, or even you could say, which aspects of the drudgery do you find less problematic, right? Right. And so, and because the truth is, like, you do not want to index on the super positive, successful side, right?
Right.
Being a tenured professor somewhere is great, and starting a successful company is great, and they each have their various, perks. but the likelihood is that's not how it's going to go, right?
Right.
Of course, all of us who are doing this are delusionally optimistic that it will, but the truth is it's the odds are stacked against you either way.
Yeah.
And so the question is, what do you really like spending your time doing? And so for me, there's a lot of things in academia that were really attractive. I loved being able to just put my head down and work on a problem, collaborating with my peers. I liked mentoring, and obviously there's a ton of intellectual freedom that comes along with it.
Right.
And I think the key to like either whether it's starting a company or working at a company, the question is to what extent does what the company actually does align with your interests, right?
Right.
Because the truth is the lines are really starting to blur between what can be done in the academy and what can be done in a startup or in a small biotechnology company, provided that there is a business case.
Right.
And so I think, for me, it was like, well, it's funny, some of these questions actually have become moot, and I'll say why in a moment, but do I want to get money by pitching or by writing grants, right? Do I want to have, meetings with my team here or committee meetings in an academic setting?
Right.
Obviously there's no real opportunity for teaching. Do you like teaching, right? Or do you not like teaching? Mentoring is a big one, right? And so, but what I found is actually it's not as different as you might think. Like, for example, nobody likes writing grants. And yet, I've done 3 financings. I have written multiple grants. I still have had to write grants.
Right.
Hopefully we're sort of transitioning out of that. It's been for over 4 years now. And hopefully me writing grants is going to stop being a thing, but I've still have had to do it. Right.
Right.
That didn't go away. And at the same time, like opportunities for mentoring aren't as scarce as I thought. I've really enjoyed being able to work with you and your team, and I have some other mentoring that I do. And so you can still do it. And of course I have my team here, and we have people in all stages of their careers that, I really enjoy, talking to, and whatever guidance I can provide, if it's thought out,
Right.
is tremendously meaningful. So it's not that much of a distinction as one might have thought. And so, it's like, there are still these differences, right? You still do have to do a lot of, my primary job as always is keeping, money in the company, right? So a lot of it is fundraising. But the question is, do you like the other aspects of the job? So for example, I spend a lot of my time in what's called business development, right? So trying to like identify what is the market opportunity for what we're doing, seek out potential partners for whom we could work with, and really trying to establish the business case, building relationships. There's a lot of networking involved.
Right.
You do have to get out there and do a lot of meetings with people. And, for some, scientists, that may or may not be what they want to do. Science is still a very social endeavor in the academy, but it's just a bit different in how it's carried out. There's a lot of critical communication either way, but it has sort of a different tinge to it. There's still a lot of salespersonship in academic science.
Right.
There's a bit more salespersonship in the commercial world, right?
Right.
And so it's just a bit different in what you have to how you have to orient yourself. Intellectual property becomes a thing you have to know a lot about. If you're making therapeutics, you have to understand the regulatory side of things.
Right.
And so does that stuff interest you? Do you like learning about IP? Do you like learning about business development? Do you like learning about market analyses? And then there's sort of different organizational structures, right? Running a laboratory is one thing. Running a business is another, right? So, I do run a laboratory, but I also have a chief science officer who does most of the running of the laboratory, right? And so I have to focus on other aspects of the organization that a PI in a laboratory doesn't, right? And so I still do get to be involved in the science, and I get to, have a large say in sort of the vision and the direction, and I get to look at data, but I don't get to generate data anymore. And, I don't get to spend, 90% of my time thinking about it like you would if you were a PI of an academic lab. So, you have to think about, are you willing to make that trade-off and think about these other things a bit more? And in order for that to be the case, you have to like them, right?
Right. That makes sense. I was actually going to ask you about because I know that, or I've
I don't know because I've not worked in industry, but I've heard that like it might be a little bit more difficult for having, like you mentioned, intellectual freedom, because a lot of a big part of industry is that it's kind of driven by money. But I think that's good to hear from you, like a different perspective about, you know, if that's what you want to do, then like there is intellectual freedom there. And I actually didn't realize that in industry you still write grants. That's so there's no escaping it, unfortunately, but yeah.
Yeah, so the answer to both those questions does relate to the size of the company. So at the end of the day, any company is, you know, all about, you do have to, you know, produce a return on investment if it's a venture capital backed company,
Right.
and if you're a public company, you need revenue, right? And so, you know, it very much absolutely is the case that you have to make money, right? Otherwise, you know, you shouldn't be a company. But what I think, what I would want to emphasize is, again, this point of whether or not the company's vision and goals and focus aligns with your own. Because if that's the case, the fact that you have to make money is yes, that's true, but the sort of, it doesn't really relevant if you're doing what you want to do anyway, right? Now, it is true that, you know, the goals of the company can change, the priorities can change, the direction can change. You may be asked to do something you don't want to do. And if you work for a company and, you know, your boss tells you have to do something, then that's how it is, right? And if you're in, you know, in an academic lab, or you're a PI of a lab, you know, you have a little bit more freedom to adjust that depending on your own interest, but it's still there, right? You still have to be able to convince someone to give you grant money, right? You still have to be doing work that's relevant. It's just driven by different forces. And so that's why I come back to this notion of, you know, whatever the market forces that are driving your company's direction, if you want to be at a company, it's really a question of, you know, are the likely directions of that company aligned with your research interests, aligned with your passion? And if so, it can be a great place to be. You know, there is another aspect I would call out. And again, I'm only speaking to small companies here because I've never worked at a giant pharma. I have some sense of what it's like to be there. But, you know, my company is 16 people, and that's a recent phenomenon. We were 3 people when we started. So everything I will say from my own personal experience relates to companies.
Yeah, but that's amazing how much you guys have grown since you started. Like, I mean, and like next year, what you're talking about with like growing even more, that's like amazing. But anyways,
and it's amazing how the structure changes, right? Because when we started it, you know, I was pouring plates and pipetting, right? And we're 3 people. And now we have, you know, 3 divisions with a hierarchical structure, and it's all these things that have to happen in order to begin to really be to produce at a company as you grow. And so yeah, so anyway, I think we wanted to talk a little bit about the question of what happens with your interest level at these companies, right? Is that where you were?
Yeah.
Yeah. So anyway, so it does kind of come back to that question of alignment and growth and sort of where you're at, where the company wants to go and where you are. And if you can align those, that's really important. The one thing I was going to add though is at companies of our size, at least at our company, and I think this is true of others of our size, there's just this incredible feeling of a team mentality. And this is, you know, not to say this can't happen in academia, and it often does. But there is still this aspect of attribution of credit that has to be carefully assigned in any academic collaboration, and that can be challenging, right? And so it's like, you know, it's always this question of, you know, who's first author on the paper, whose idea was it, who's doing what, and that's just, you know, there has to be some sort of currency in order for people to advance their careers. There's no getting around that.
Right.
One of the things that you find at a small company is that if it's done correctly, that kind of goes away. And what you've got is, you know, a small group of people who all are there for each other, right? And very much sink or swim together. And if everyone buys into the vision and the culture, and your culture is sound, you know, it can be a lot of fun, because that, you know, there isn't really any question of who gets credit for what, right? People are compensated better economically than they are in academia. They do have some ownership of the company, right? So they care about financially the success, but they're also just beyond that is this feeling of like, hey, we're a team, and I want to succeed so that you succeed.
Yeah.
And, you know, I would I think one of the most gratifying things we've heard as we try to like sort of anonymously get feedback from our people is that's been true for them. Like some of the folks that we have who, you know, came directly out of postdocs have told us, like, this is one of the things I love about Grow Bio is, you know, that feeling of camaraderie, and, you know, there isn't really a sense of competition. There's a sense of, you know, we're here for each other, and, you know, very much wanting to win for the person standing next to you.
Right.
So that this isn't, didn't mean this for to be sort of like a selling point necessarily, but I do think that is a difference, that can be a difference that's worth considering. And again, it's not always the case that it's a problem,
Right.
but it is a feature that can happen, again, if you have the right alignment between your research interests and a small company's research interests.
Right. And I was actually going to ask you a little bit about employee well-being and how I imagine that everybody kind of through COVID may have had like a more difficult time, but I'm just curious, like what you do as a CEO to be able to support your employees and to kind of help motivate them and encourage them through like maybe difficult times, or just as a company overall, adding on to the team mentality.
Yeah, it's a great question. I mean, from the management standpoint, a few important things. One is sort of around transparency. And so it's really important, I think, for folks to feel like, to a certain degree, they know what's going on, and that, you know, you have their best interest in mind. And it's an interesting line to walk, because between any in any organization, you know, there's an executive team and sort of, you know, in this case, there's an R&D team we're talking about. And the R&D team, you know, sort of generally, they can't know everything that's going on, and probably shouldn't, right? There are vacillations in what's going on in any company at the executive level that the whole company, quite frankly, needs to be protected from, right? It's not something that people, you know, there's levels of, you know, that would be relatively stress inducing that not everybody needs to know about. And that's not speaking just to our company. That's true in every company, right?
Yeah.
On the other hand, if you're too opaque and people feel like there's stuff going on they don't know about, that's even worse, right? And so they'll start to feel like they can't trust their management, and they have to worry about things. They have to maybe they create their own sense of what's going on that's detached from reality, right? And so for example, you brought up COVID, right?
Right.
So at the beginning of COVID, which was, you know, not quite, you know, so in March it'll be 2 years ago, March,
Right. When
things really got bad. And so we had an all hands meeting where we, you know, wanted to acknowledge what's going on. We basically wanted people to understand, you know, where we're at as a company, you know, where we were in terms of our financial health. We were fortunately in a position where we could say, like, you know, we're not in any sort of imminent danger. We'll, you know, but, you know, we do need to sort of monitor the situation and see how things proceed, and try to basically give people a rough sense of, you know, where we stand. But then we also met with each person individually and wanted to sort of get a sense for, you know, what are their own personal concerns? What's their level of comfort? How do we build a policy around how we should work together going forward? And that's sort of generally how we tried to run things here, right? Really, because we're small enough to really talk to people, you know, sort of as a group, but also individually, and build that feedback into how we run the company as best as we can. And so that brings, I think, a couple other really important aspects of making people feel comfortable and happy is around not only just transparency, but sort of incorporating their feedback and also acknowledgement, right, of what they've done. People want to feel like they're heard. People want to feel like they've been acknowledged for their successes, and they want to be supported when something hasn't gone as well as it could, right? And so that's the kind of culture you really try to build early, and that you fight fiercely to protect. And that's part of why it's so hard to hire when you're smaller, because every person is so important to maintaining that culture. And eventually, there's no denying it. It gets diluted, and you can't keep everything that you have as a 10, 15 person company, as a 100 person company, if you're lucky enough to make it that large. But those sort of aspects around transparency, communication, camaraderie, acknowledgements, and incorporation of people's feedback are really critical to sort of building and maintaining that culture. And then also, fun, right, is important. And you want to sort of encourage and nurture that. Like, so for example, at our company, there was a movement after we closed our Series A, we started building out what we call a high throughput foundry, which is this collection of all these robots and automated liquid handling systems that allow us to accelerate the process of bringing new amino acids into our platform. And so there's a lot of machines that were ordered and brought in, and there was this movement within the company that every instrument would be named after a Pokemon character. Now, probably the greatest sacrifice I've made to the company is to accept this, because Pokemon is sort of after my time, and for me it would have been Simpsons or Nintendo characters, and I had to let them go with Pokemon, and I did it. It was a tremendous sacrifice.
Gotta take them for the team sometimes.
Really important, yeah. So I don't know what any of these instruments are. I don't know what they are. I presume they're all appropriate. There's no HR violations in there, but it's great, right? I mean, we have a whole Slack channel set up for Pokemon names for the instruments. And during COVID, it was difficult to have sort of team bonding exercises, but prior to that, and now we're finally getting back to it now, really finding time away from the laboratory to just hang out in something that's in non-work related environments, and build that sense of enjoyment of each other's company. I mean, I feel very confident that our team likes each other, they like being around each other, but you can sort of foster that more outside the lab, and we've tried to work hard to do that, both through sort of just kind of fun activities, but also trying to work towards some service projects that we can do together as well.
Right.
So those are all things that we're trying to sort of hopefully accelerate now that we're back into in-person living.
Right. Yeah, that's really great. Actually, I really liked a lot of the points that you made, because I know that, the point of iGym is to eventually have like a spin out and do like a startup company from a product that you make or are starting to develop. And I think I kind of got a little bit of an experience with running like a team this year as like president/vice president. Like Lindsay was our president, and then I was like the vice president, but I kind of did a lot of stuff more so for like wet lab, HP and math modeling, whereas she was doing more stuff with administration work and like organizing the team. And I think that it can be challenging, I think, to like get to that point where you guys are all like working like as a cohesive unit, especially when other people are doing different things and you're not around each other all the time. But I think a lot of the points that you made, maybe we can implement next year with our team. And then, I had one more question I was hoping to close out on, which is, so you've talked a lot about like the work you do and, running a company. And I'm just kind of curious as to what some of your hobbies might be outside of lab or outside of your company, just to kind of relax or, doing stuff away from that, like what you like to do.
Yeah, sure. So I guess it's getting colder now, but like when it's warmer during the summer, I don't know, I guess like 6 years ago, I took up sailing on the Charles.
Oh, okay.
So we're lucky enough to live like near water here. So
yeah.
So I've been doing a bunch of that. I also, before COVID, I captained a co-ed slow pitch softball team that was actually not dominated by scientists, believe it or not. So as far as like getting away from work, it was a really fantastic way to like have something that was completely orthogonal to everything I would do all day. And so hopefully I can get that team up and running again this summer. And I know some of the folks here have some talent or talented spouses. And so there's a chance maybe there'll be a Grow Bio team, maybe there'll be my old team, we'll see. I like to ride motorcycles. I had to sell my motorcycle when I moved to Boston, but I recently did a trip in Vermont that was really fun. You can still rent.
Wow, awesome.
So I do some of that, and then when it gets cold, I tried to pick up some new hobbies. I'm trying to get into bonsai. This making tiny Japanese trees.
Right.
So I have my first bonsai tree that I'm working on. And actually, I was mentioning like when I was a kid and I tried to get into I was interested in special effects. I was really into stop motion animation, which is where you kind of frame by frame, claymation as an example, or like with little Legos, take one frame, move it a little bit, take a frame, move it a little bit, and create little stories that way. This is super easy to do now on an iPhone, right? There's like free apps where you can do this way better than I used to do with my 47-pound video camera. And so something when it gets cold, I'll probably get back into that a bit. Make some more stop motion animations, and music is another hobby of mine. I was a drummer for a long time. I love to go see live music, and don't really play anymore, but I mess around with some friends sometimes, and so that's hopefully going to be perhaps revived a bit as the winter goes on.
Were you in a band? Like
several. Yeah.
You were in several bands. Wow, awesome. That's super cool.
Well, anyone who's been in one band has been in several
bands.
Yeah,
fair enough.
Yeah.
But yeah, anyways, I want to say thank you for meeting with me and for being willing to share your story and to share some stuff, kind of information about the work you do now. This was a really fun conversation for me.
Oh, likewise. It's my pleasure, Lizzie, as always. And on my end, it's been great to see the progress of your team. I think you guys have been a great job managing it. So yeah, I'm really thrilled to sort of stay involved and see where you guys go next.
Yeah, thank you.